Okay, so hello everybody. I would like to start by thanking the organizers for giving me this chance to talk to all of you about the work that I've been doing recently at the University of York, in particular about Moorhen. And I would like to start the presentation by actually talking about what is Moorhen. So Moorhen is a next generation web-based application for the visualization and manipulation of molecules in a structural determination and analysis. So that's essentially the official sort of description of what Moorhen is. But I guess if we wanted to simplify it in short, it's just Coot on the web browser. So I'm going to assume most of you are aware of what Coot is. Coot is a desktop application that essentially will let you load a map and, well, multiple maps if you want, and a series of molecules. And then you can do things like interactive model building, experimental validation, etc. So what Moorhen aims to do is just to deliver that same set of functionalities, that same experience on the web browser. The way essentially you would start Moorhen is different than the way you would start desktop Coot. So essentially you would just open your browser of preference, of choice, so Firefox, Chrome, or Safari, type in a URL, and then you essentially get started using Moorhen without the need to having to install any software. From the technical point of view, we take a cross-section of how Moorhen actually looks like. It will look more or less like this. So sitting at the core, we have what we call the Coot API. This is just a set of C++ libraries that essentially these libraries are shared with desktop Coot, and these are the libraries that are in charge of performing most of the calculations. So things like model building, refinement, validation, those calculations get done at that level. And because this is shared with desktop Coot, it means that if you were to perform an action in Coot, so say for instance, model refinement, and you do the same thing in Moorhen, the result is going to be pretty much the same thing because it's the same code being executed performing that calculation. Now, the difference is what we do in Moorhen, we take that Coot API that is essentially headless, so it has no UI, and then we build around it a UI that is built using React, which is just it's a framework for building web-based UIs, and that's what you use to actually interact on the browser with the Coot API. Another technical aspect that I think is important for users to understand is that the app is 100% client-side only, and what this really means is that when you're using Moorhen, you're using your laptop or your machine, whatever you're running Moorhen, and essentially everything is happening in the CPU of your machine, right? So we don't actually have a server. There's no communication required with the server. In fact, after you load the page, you could just turn on airplane mode, and even if you, essentially you wouldn't even notice that you have lost internet access. You could just work with it normally. And what that means as well is that it's 100% private, right? When you are actually, say, uploading a file into Moorhen, you're not actually uploading anything. You're telling the web app where to find that file in your file system, but everything is happening in your laptop, which again just means 100% private, and it's blinking. And if it's that is especially important if you're working with essentially with sensitive data, right? So you don't have to worry about that. What is the current state of Moorhen? So, well, the original motivation of the project was just to offer Coot users the ability to use Coot in any device at any time. So regardless of OS or software updates, or whether you're using Mac, Linux, Windows, whatever, we still want people to be able to use Coot. And well, after intense development, I'm happy to say that Moorhen is in a state where it is routinely useful in the most common day-to-day tasks. In fact, Paul Emsley wrote a list of sort of all the must-have features that would have to be in Moorhen before the release, and all of those features are now available in Moorhen. In fact, Moorhen extends on that original list of features. That's the list of features that he wrote. Essentially, there are three categories. We have model editing features, so things like peptide flip, adding and deleting residues, dragging atoms, refinement, etc. We also have presentational features, so SSM superpose, map contouring, also being able to change the color of the map and the molecules. And we also have validation features, so things like density fits, row timer validation, geometry analysis, etc. But then again, that's just a basic list. The truth is, Moorhen, as of today, extends on that list, and I just couldn't fit all the features all of the things that you can do right now in Moorhen, so just keep that in mind. Also, something important as well to say is that Moorhen is also intended to be a web-based replacement for CCP4MG. So CCP4MG is a program by CCP4. Essentially, it's a program that you can use to generate pretty pictures, essentially, for publications. So there are also a series of figure-making features on that list of Moorhen features. So things like shadows, depth blurring, ambient occlusion. We also have basic movie making. But then again, that's just the basic list. Moorhen actually extends on that list of features. And to sort of make the point, I have a few slides with a few pictures that I've taken on Moorhen. Its first picture is a cryo-EM map. It's colored using the output of Occupy. So essentially, dark blue would be high occupancy, and then it goes all the way to yellow for low occupancy. But what I really want to show is that, well, essentially, picture right now is completely flat. It's difficult to tell what's the actual shape of the map. You don't really know what's inside the map, what's outside, and that's because I've turned off ambient occlusion for this picture. But we do have ambient occlusion in Moorhen, so if you turn it on, then it will essentially look like this. So this is the sort of picture that you can take in Moorhen once you turn on ambient occlusion, which is obviously very important for cryo-EM. You have a better idea of how actually the map looks like, and the picture becomes much more informative. We can also look at big molecules as well. This is a virus capsid, but again, ambient occlusion is off, so you cannot really tell the shape of the capsid. All you know is there's just something on the shape of circle. When we turn on ambient occlusion in Moorhen, then you get this sort of nice picture. So yeah. And you can also look at small things as well. So for instance, you could be looking at a ligand like this thing here. So this ligand, I'm using a representation, is some sort of blob representation colored in yellow. And then we have the structure in these blue ribbons. And there are several things going on. Actually, we have depth blurring as well. So you may notice that the beta sheet on the back is sort of blurred. That's because it's far away. So that helps the user give this sort of perception of depth. And then we also have shadows being cast by the ligand on that beta strand on the back. So there are several things going on that make sure that the picture actually looks nice. Right, so now I have a series of video demos. So these are just videos I have recorded myself using Moorhen. Let me just put this full screen. Yeah, so I essentially have recorded myself using Moorhen. I'm just going to talk over. This is Moorhen running on Chrome on my laptop. And all I have to do is I just have to go to moorhen.org and essentially just have Moorhen at my disposal. This is how it looks like when it starts. It's just a white screen because I haven't loaded any data yet. The UI is very clean. We only have one button, which is that hamburger menu on the top. And when you click on that, essentially they will have a drop-down menu. This is essentially the same items that you will have in the top menu bar in Coot. So things like file, edit, calculate, etc. That's essentially where you can find it. And the first thing I'm going to do, I'm going to load some data into Moorhen. So I go file, and then I could upload something from my laptop. I could fetch from the PDB, but all I'm going to do, I'm just going to load tutorial data. And that will essentially bring in a model, a map, and a difference map. And I'm going to try to reproduce the workflow, typical workflow of somebody using Moorhen, which is essentially you're given a model and you're given a map. And you try to figure out what's wrong or what could be improved in that model. So I'm going to go to validation, and you can see there the list of things that you could potentially do in validation. We don't really have time to talk about all of them, so I'm just going to go to Ramachandran plot, and that will open a window that I can just drag around, and I can resize it as well if I want. For instance, I want to look at bigger Ramachandran plot, so I'm just going to make it bigger. And everything is interactive. So I hover over points. Essentially, Moorhen is telling me what I'm hovering over on that label on the top right. I can also click on things, and that will essentially center the view on that point. So for instance, I'm going to click on that outlier, and that will center the view on that outlier. Now, looking at this outlier, what we can tell is that there's clearly something wrong going on. That oxygen atom is sticking out of the density, and we want to fix it. Now, if this was desktop Coot, what I would do, there's a site, there's on the side, there's a menu with a series of buttons. So I would click on peptide flip, click on the residue, then Coot would flip that residue, and then essentially the oxygen atom would just go into the density. We don't have that sidebar anymore. Instead, the way you interact with things, you simply right-click on whatever you want to modify. So if I want to modify that residue, I just right-click on that residue, and that will open a context menu with the actions that I can take on that residue. So I'm just going to right-click, and then I'm going to click on flip peptide, and that will just flip the peptide. So the same thing again with another oxygen atom. I'm just going to right-click on it, and instead of flipping the peptide, I'm actually going to drag it. So I'm going to click on drag atoms. I'm just going to pull the oxygen atom up. There you go. And now I'm just going to click accept, and now it's fixed. So that's just very quickly some of the stuff that you can do in Moorhen. Again, I don't have time for everything. But you can also use, yeah, obviously that's sort of the kind of work that you can do in Moorhen at the small level. So looking at the small details, but you can also look at the big details, right? So at the big level. So this is Moorhen running in Doppio, which, well, it has been released today. So Moorhen is in Doppio 1. And I'm just going to go to jobs. I have import job where essentially I have imported a map. So you can see there the map. If I go to IO, I can select that map. And then there is a Moorhen button that if I click, that will essentially just launch Moorhen inside Doppio with the input map. Right, so the first thing that you can see is that the map is a bit noisy. So I'm actually going to blur the map a little bit. So I'm going to go to map tools, and I'm going to blur it. Yeah. And now I'm going to go to the Maps tab, which essentially is a menu item that lets me control all of the display aspects of the map. So things like contour level, radius, etc., all that kind of stuff I can change there. And I'm going to make that map a semi-transparent surface, right? Because that's good, what you would typically want to do with these things. And now I'm going to bring in a model, which is in my file system. So I just go File, Coordinates, and I'm going to upload that file. By default, Moorhen will draw it as bonds, but I can just change that to ribbons using the Models tab. So this is just like the Maps tab. It lets me essentially control display settings of the maps. You can see bonds are active, but I can turn that off, and I'm going to turn on ribbons. There you go. And now, just looking at this, you can tell that there's something going on. There is a part of the map that is lacking the models that has not been modeled. We can take a better look if I, for example, I create a mask. So if I go to Map Tools, Map Masking, and I'm going to create a mask for that map using the molecule. Yeah, so now we clearly see that there is indeed something missing. Now, this is the post-AlphaFold world. So I know for a fact there's this AlphaFold model in the AlphaFold database for that chain. So all I have to do is I go File, then Fetch from Online Services. There's a drop-down menu, so I switch from PDB to AlphaFold database, put the Uniprot code for that chain, and I just fetch the AlphaFold model. I'm going to display that as ribbons now. And by default, these models from AlphaFold are colored with the PLDT color scheme. So essentially, dark blue is good PLDT, and then it goes all the way to orange for low PLDT. And now I'm going to try to center the view on the masked map. And now, of course, the challenge is to fit that molecule into the map. And of course, what I could do, I could just simply move the molecule there. So I could just go Edit, Move Molecule, select the AlphaFold model, and then click OK. But that's not what we want to do, because that's just going to move the molecule, whatever I'm looking at. What we really want to do is a form of rigid body fitting, which is in Moorhen is called Eagle Fit with Fourier filtering. And that will actually just perform rigid body fitting, and it will try to fit the AlphaFold model into the map, which is exactly what we want to do. I'm just going to increase the number of trials because I really want it to work. Click OK, and that's going to take a little bit. It's actually taking a minute and a half, which is more time than we have. So I'm just going to skip forward. Should be somewhere around here. Yeah. So it takes a while, but it does a good job. So as you can see, it fitted the model. It fits very well. A few things that you could do now, for instance, you could try to refine that helix, or maybe try to chop out the parts that have low PLDT, but essentially we're done here. So I'm just going to go Edit, Merge Molecules. That will merge my fitted AlphaFold model into the original model. And now, because we're in Doppio, there's that Save button on the top. So I can just click on Save, and that will essentially save my molecule into Doppio. So it will just export it back to Doppio. If I go now to IO, that will show me the original input map and also the output, which will be the PDB files, essentially. And from then on, I can just do, essentially, we could just keep working in Doppio with the output from Moorhen. Other things as well. So this is the last demo. Let me stop this. Quality. Right. So refinement with restraints. This is, again, a similar situation to what we had before. We had a map and a model. The model sort of, it's different. Well, I mean, it fits the map, but it's different because, as you can see, the model doesn't really fit the map that well. So there's that helix in the center that doesn't really fit. We have this helical pair here as well that doesn't really fit. So what we need to do, we need to refine the structure into the map, right? But ideally, we would also like to use distance restraints so that the overall structure, the shape of the structure is kept. So that's exactly what we're going to do. We're going to go to Calculate, Generate Self-Restraints. And now essentially what Moorhen is doing, it's taking all of the atoms in the structure, and then for each atom takes all of the atoms within 6 angstroms and generates restraints for that atom pair with those distances. And that's what you see there with the thin gray lines. And those restraints are then taken into refinement, which is what I'm about to do. So I'm going to create a selection that's just selecting the whole molecule, because that's what I want to refine. Click on Refine, and that will essentially refine the molecule. So the refinement in Moorhen is animated, so you will actually see the molecule sort of moving around and fitting into the density with each cycle. But then again, this takes a little bit too much. It takes a minute and a half, so I cannot really show it. But you would be able to see it essentially just sliding into the density. And when it's done, you are presented with this sort of, well, with a lot of green and purple, which is essentially those are the same gray lines that we saw at the beginning. But what happens is as the atom pairs, the distance between the atom pairs deviates from the target distance, then we color those restraints from green all the way to purple. So that essentially, I mean, that is to be expected because, you know, we did refinement, but the important thing is that the restraints did their job. The molecule fits a lot better now than the map. If I clear the restraints, you will get a better view of the molecule. I mean, obviously, it fits the map pretty well. It did break the geometry a little bit, but we are in a much better place now to start working with this molecule now that it is actually in the map. So there are obviously other things that you will want to do from there. Right. So you've seen basically a taster of what you can do in Moorhen. There's obviously more and more features that we're adding, and we're working hard to add more and more features. We're also having collaborations with different groups to generate different plugins for Moorhen. This is, for example, a very recent plugin. Oh, you cannot see it. Okay. This is a very recent plugin that we have added into Moorhen. It's a slice and dice. So it's software that you might be aware of. You have maybe heard of it. So it's software by Rigdon Lab in the University of Liverpool. And essentially, we have it working in Moorhen. You have a nice UI where you can essentially run a slice and dice within Moorhen. Where is Moorhen available? So as you've seen, it's available in Doppio. There's the Moorhen button that you can simply just click, and then it will open Moorhen. It is also available as part of CCP4, so it's in CCP4 Cloud, and in the future, it will also be available in CCP4i2. It's also available as a standalone application in moorhen.org, which is probably the easiest way to access it, but then you obviously lose all of the ecosystem of CCP4 and CCP-EM, so I would recommend using Cloud or Doppio. Also available as a standalone application as an Electron app. And then we also have it as a React component, but this is essentially for developers that may want to introduce Moorhen in their app. So for instance, this is what happened. This is work by Jordan at the University of York. They have a privateer web server. And essentially, as I said, we offer Moorhen as a React component, so you can just insert it in your service. And in this privateer web server, essentially, they just put a small window where you can look at the output using Moorhen, and yeah, they have Moorhen running there. Right, so I'm just going to finish now by thanking the Moorhen team. So that's Stuart, Paul, Kevin, Eugene, Jacob, Martin, and Lucrezia. Thank you.